Okay, good morning. Welcome to the plenary session on quantum biology. And we have three excellent speakers today. Tonight is the party. We were going to have it outside, but we couldn't do that, so it'll be down on the third floor. And there'll be food, beer, music, and a poetry slam. And I'll announce this later. If you want to give a poem, I'll ask for a show of hands, and we'll see how many we have roughly, and therefore what time. We'll start, but probably 7 to 9, then music and dancing to follow that, and until the beer runs out anyway, and we'll take it from there. So in any case, it's my great pleasure to introduce the speakers today. Our first speaker is someone who made a big splash this year. He's made splashes before. With an amazing paper on SuperRadiance in tryptophan networks in cytoskeletal structures. He's Philip Kurian from the Quantum Biology Lab at Howard University in Washington, D.C. And his topic is Super Radiant Life from Slime Molds to the Stars. Philip Kurian.
Thanks, Stuart. No, thank you so much, Stuart. Thank you to the organizers who put on this great conference, to Travis and to many others who, I haven't interacted with personally here. But thank you all for coming to this early session. I'm really excited to share with you some of the work that we've been doing in my lab in Washington, D.C., the seat of empire, so forgive us for our sins. I am very privileged to be the principal investigator and founding director of the QBL in Washington, D.C. And this is a work that is really the fruit of the last decade of our efforts to both theorize, simulate, and now experimentally validate these quantum optical effects that are occurring in many biological systems, not just neurons in brains, but even in aneural organisms. And so what I hope to give you today in the time I have is a flavor for this work, but if you like to see more details, there's a lot of references, some starting even earlier than 2017, but here's the thread of this line of work in my lab. The last three in rapid fire came in the last 2 years, year and a half, and as Stuart mentioned, this has given us a lot of press around what SuperRadiance could be doing for information processing in both the cell and in the brain. So I'd be remiss if I didn't thank all the amazing people with whom I get the opportunity to work, and without whom my work would be absolutely impossible. So I'm going to highlight two groups amidst all of our support members and collaborators. One is funded by the Alfred P. Sloan Foundation and involves our intern coordinator and administrator, James Murray, and my postdoc, Suyash Bajpai, but collaborators at Tufts University and at Amoeba Energy in Japan. And then the SuperRadiance work was by two postdocs in my group, a very talented undergraduate student, and our colleagues at the University of Florence and at EPFL in Switzerland. And that work was funded by the UK-based Guy Foundation. We also have simulation resources at the Argonne and Oak Ridge Leadership Computing facilities. So here's the problem for a theoretical physicist studying quantum biology. We have a zoo of complexity within even a single atom. As all atoms are ensconced in the electromagnetic field, photons and antiparticles and particles can come into and out of existence on timescales that are far beyond our capacity to measure. And even this picture is somewhat of a mental map, but it's a bit of an untruth, right? Because the nucleus, too, is not these balls and particles that you're seeing, but it is fields. It's fields governed by QCD and quantum chromodynamics. And the photons that you're seeing coming and the electrons that you're seeing are governed by interactions that are governed by the electromagnetic field. So how do we reduce that complexity without coarse-graining away all of the quantum degrees of freedom? Well, the theoretical physicist's answer is a two-level system. So we take an atom, you take a molecule, you take any sort of object that has all this complexity and degrees of freedom, and you can reduce it more or less to a two-level system, which can absorb a photon and emit a photon. And so this is what I'm going to talk to you about today, but in the context of, do we have video? Is that me? Oh, maybe it's my laptop. Okay. Okay. So sorry. Yes. I am. That's very strange. A moment. I had power. That's weird. Yep, about to do that. All right. Okay, so where were we? All right. So reducing this zoo of complexity, right? How do we reduce this zoo of complexity without sacrificing meaningful quantum degrees of freedom that can actually manifest and be observable at a mesoscopic or macroscopic scale? So this is a bit of a state-of-the-art question, but we can reduce it to something that anyone can understand. So we want to look at observables of these collective interactions. So if I take a two-level system that I can treat and call as a qubit, I'm going to write that suggestively as 0 and 1 here and 0 and 1 here. But now I put them in interaction. So when I take two qubits and I put them in interaction, I'm going to get aggregate states. I'm going to get what I'm going to call an upper state, a superradiance state, and a lower state called a subradiance state. This is also what you would see if you put oscillators together in a classical context. But here, these are optically excited aggregate states, okay? So now there are spectroscopic signatures of those aggregates. So what we would normally see is that center frequency. That is a quantized resonance that occurs because of a discrete eigenspectrum between the 0 and the 1 state. And that 0 and the 1 state is that center frequency. But now these superradiant and subradiant states are red shifted and blue shifted accordingly, and you can see that spectroscopically. So we have a collective observable that we can see of these aggregate states and of these interactions. And they emerge fundamentally from the interaction of quantum matter, quantized matter, discrete energy levels with the electromagnetic field, or the quantum of the electromagnetic field, the photon. So now, what happens when we do arrays of these two-level systems interacting with and in the electromagnetic field? So this can be very suggestively written in this form of the wave function. So I can no longer just have a single photon in a single molecule, which would give me, if I have n chromophores, I would need n photons to fully invert the ensemble. But here, I can take even a single photon and I can share it coherently across the collective. So I can put the single photon in the first chromophore, I can put it in the second, I can put it wherever I want, right? But a coherent superposition is what the wave function would be in a quantum context. And many groups, not just my own, including Marlon Scully's and Anatoly Svidzinski's and Ana Senjo Garcia's, have investigated this for the last 2 decades. This phenomenon of single photon superradiance in both collectives of shells and in arrays of quantum emitters in various contexts. So our group, my group, is the first to kind of apply this to living matter and to biological arrays. So this was the work that got a lot of press last year on ultraviolet superradiance from meganetworks of tryptophan. And so this is Gustavo and Nathan, two of the postdocs that did the work. And this is our colleagues at EPFL, Majed Chergui, and at Florence with Giuseppe Luca Cilardo. And so this got quite a bit of press from Science News with Sabine and PBS Spacetime. But I do want to caution that this has no support in this or overlap with orchestrated objective reduction theories. However, there may be some other curiosities that are fundamentally quantum that are emerging from this research. So what we show here, and what I'll present to you the experimental data for, is a collective observable that demonstrates that quantum effects actually can rise above the thermal noise floor, and we don't need to invoke gravity or anything exotic, just the fundamental force that is operating in biology, which is the electromagnetic field. So imagine, many of you are biologists, I'm assuming, and some of you are physicists, but these blobs that you see on the screen in proteins are ubiquitous in biology, but they form with these architectures of tryptophan. Tryptophan is the most fluorescent amino acid in naturally occurring biology, and being such, it absorbs an ultraviolet photon and emits at a slightly longer wavelength. So these architectures of tryptophan occur not only in microtubules and in actin filaments and in amyloid fibrils, they show up in transmembrane proteins like G-protein coupled receptors. They form in photoreceptor complexes in plants like UVR8. They're in SARS-CoV-2. They're in cellular organelles and appendages like flagella and cilia. And so here you can see the tubulin protein, which actually has, let's see if I can, yep. This is the tubulin protein, which has 8 tryptophans, 4 in one monomer, 4 in the other. You've got in a micron of microtubules about 10,000 of these tryptophans, and then you can have hundreds of thousands and millions, and even more in structures like the flagella. This is called the axoneme, which is the basal body of the flagella and the cilia, and the centriole, which helps orient the cell towards different electromagnetic stimuli. And it's one of the most evolutionarily conserved structures in all of biology. And of course, on the right, those were axon bundles of microtubules. So here's the experiment that shows, it's a very simple experiment, has to be done very carefully and quantitatively, but it's a fluorescence quantum yield experiment. So the idea is as follows. We can excite these architectures throughout the ultraviolet, but the only molecule that absorbs in proteins at 295 nanometers is tryptophan. Above 295, tryptophan is the only molecule that absorbs in the ultraviolet above 295. Now, others absorb in the visible and elsewhere in the ultraviolet. But here, I want to show you our prediction. This is a prediction using an open quantum systems approach. If you're a specialist, you may know the Lindblad master equation. So we've used the Lindblad master equation to derive radiative decay rates for the collective ensemble of quantum emitters, and then computed the fluorescence quantum yield, as you can show here. And what everyone expected before we did the experiment was that you'd just see the same thing in the experiment that you see with tryptophan in solution, the same 12.4% that you see at the bottom of this slide for a single tryptophan in solution. But in fact, that's not what we see. So at this quantum yield at 295 nanometers, where only tryptophan is absorbing, I want to draw your eyes to the tryptophan in solution, which is 11.4%. And what's interesting is that when you go from tryptophan in water, or in buffer, to tubulin in the same buffer, you put the tryptophan in the protein context, the quantum yield goes down. Now, why is that? Well, that's because the non-radiative channels in the protein increase, and so the quantum yield goes down. Now, what's intriguing is that as you go in the protein from tubulin to building a micron or 2 of microtubule, then you have the radiative decay rate enhancement that swamps the non-radiative decay rate increase that lowered the quantum yield in tubulin. So this is really a smoking gun that this, and we've corrected for all scattering effects, so that's not an issue here. So the fact that superradiance is so dominant in this case suggests we have a good observable that that is robust in, again for specialists, the thermal Gibbs ensemble, where these superradiant states are in the low energy part of the manifold and thus are weighted very strongly in the thermal Gibbs ensemble. And so this is an unambiguous and remarkable prediction for a microtubule at 1 to 2 microns that agrees with experiment within about 0.2%. Okay? So again, these architectures occur not just in brains and in neurons, but they occur in aneural organisms, they occur in single cells, but on the right, of course, you see these nice bundles of microtubules that form in axons, which of course are highly stable over decades and mammalian lifetimes. Oops, went back. Okay. I want to emphasize that this point of superradiance is really a product of the helical symmetry of protein fibers. It's not unique to microtubules. We actually showed in this work in Frontiers in Physics, again, the first author is a very talented undergraduate intern who just started graduate school, but this, Hamza, what he showed and what we showed is that this effect is a result of these helical architectures. So you can kind of imagine, even though you'd have cancellations of these transition dipoles as you go around this helix, you have a sort of enhancement longitudinally along the helical axis. And so not just in microtubules, but you see the high density of tryptophans in red in amyloid fibrils in mouse and human, as well as in actin filaments here, which is not as dense as in microtubules, but it has a different function. So these are robust and kind of multifactorial objects that do many different things besides sense and respond to light. So if you want to see, this is again a non-Hermitian effective Hamiltonian. So it's because it's a non-Hermitian open quantum system, it has complex eigenvalues, and we are plotting here the imaginary part of the complex eigenvalue, which is the superradiant enhancement on the y-axis. And you can see again for amyloids, it increases a great deal. And here's what the spectrum looks like if we take the real part of the complex eigenvalue and the imaginary part, and we plot them against each other. And you can see here how the superradiant states populate in the low energy part of the spectrum, and these are weighted very strongly even in a very thermally turbulent environment. So even though you have all this bombarding that's going on, even if you have a few superradiant states that are extremely low energy, their collective Lamb shift is highly negative. Remember, the Lamb shift is what came in the hydrogen atom, but here we're talking about a collective Lamb shift due to the collective of quantum two-level systems interacting. And because of that collective Lamb shift, we can see a very robust observable that we are currently predicted in human amyloid, and we're currently doing experiments right now to see if we can validate this prediction as well as the one that we did for microtubules last year. Because this would then open up a zoo of possibilities for treatments and biomedical interventions that involve not only light at this level, but reconceiving hypotheses about the amyloid cascade hypothesis that have plagued the medical profession for the last few decades. So if you want some intuition for where this is coming from, I said helical, but you may also understand that this is also a product of the chirality of biosystems. So here, if you take just a simple model of like all those dipoles in parallel, you don't see this effect. In fact, you see all the superradiant states in purple at the high energy portion of the spectrum. But if you want to look at this chiral object, it is very similar to what you're seeing here, and it's in the low energy portion of the spectrum, and consequently you see this rise in the fluorescence quantum yield that I showed you with the microtubules and predicted in the amyloid. So as I mentioned, the amyloid cascade hypothesis, which has dominated Alzheimer's disease research for the last several decades, this hypothesis suggests that the accumulation of amyloid in the brain is the hallmark of dementia and neurodegeneration. Well, what we are realizing is that because of these architectures of tryptophan, tryptophan has what's called a very strong absorption-emission Stokes shift. So in other words, it can take ultra-weak, ultraviolet photon emissions And these superradiant networks can superabsorb these photons and re-emit them at longer wavelengths, safer energies that are not as damaging. Still in the UV, but then at lower energies where other chromophores can absorb them and down-convert the energy. So at early stages of dementia, Alzheimer's amyloid is actually photoprotective. It can be photoprotective in terms of responding to the pathogenic environment that has a lot of these free radicals and electronically excited species that emit, we've known since the early 90s, these electronically excited species emit ultra-weak, ultraviolet metabolic photon emissions. These were experiments done by Quickenden and Tilbury with single photon detectors in the early 90s. So this was a work we did with Travis in 2017, showing kind of this theory of how ultra-weak ultraviolet photon emissions can actually be used to stimulate and signal not only along microtubules, but along any architecture of these types of chromophores. And so I refer you to my Science Advances paper, where I've taken this to its full-out extent, applying thermodynamics, quantum mechanics, relativity, and this result of single photon superradiance to understand the implications for information processing in both aneural organisms, neural bundles, and the relationship and comparison to quantum computing architectures in modern state-of-the-art quantum computing implementations. You may know the name Jacob Bekenstein. He was of black hole fame in the early 80s. And he once theorized that the ideal digital computer operates at 10 to the 15 ops per second. Of course, now we have exascale computers that are networked and go beyond that. But it's very interesting, and what I'll motivate today is that by using something called the Margolis-Levitin theorem, I can show that these bundles and these aneural filaments, protein filaments that have superradiance, can operate within 2 orders of magnitude of the Margolis-Levitin limit for ultraviolet excited states. And not coincidentally, that limit for ultraviolet is 10 to the 15 ops per second. So here's the standard model of the brain that many people, probably not at this conference, but at many places in the world, operate in classical neuroscience. And it's really important to understand. You know, it took us 20 years, us as a scientific community, to understand and map the Drosophila brain, just the classical neurons. And so these are chemical action potentials, Hodgkin-Huxley action potentials. But we can go deeper into the quantum realm, and we can understand this is 9 orders of magnitude faster than the Hodgkin-Huxley neuron, what's happening with these superradiant states. So these architectures, you can think of them as modulating the signal of the Hodgkin-Huxley spike. So within that Hodgkin-Huxley spike, it's not that you have a continuous wave of these superradiants, but you have pulses, bursts within that can be integrated above the thermal noise floor. And in fact, the thermodynamics checks out. If we compare it to the Landauer bound and we look at that limit, we can actually see that, in fact, the brain doesn't overheat even with these energy inputs. And it's in a reasonable energy throughput that isn't, for example, exhausting the rest mass energy of the brain in a very short amount of time. So what do those timescales actually look like? Well, we simulated these all the way from a single microtubule and even single amyloid and actin filaments all the way to these large structures. And when you're doing quantum simulations or you're diagonalizing large matrices that are huge, these things take a long time computationally. So I'm very skeptical when people say, oh, we can do everything quantum for every quantum degree of freedom, because we've had to coarse grain appropriately to deal with even these reduced quantum degrees of freedom in these architectures. So this is a picosecond. So that's 10 to the minus 12th of a second. So very fast, 9 orders of magnitude faster than a Hodgkin-Huxley spike. And all of these are within 1/10 to 1 picosecond, on the order of a few picosecond, for all of these architectures of tryptophan in these structures. Now, there's also, so when you have these superradiant states, you also have subradiant states, as I mentioned at the beginning of the talk. So those subradiant states are also collective, quantum optical, and they are photoexcited, but they're essentially trapped and live for very long times. As you can see, these are on the order of seconds. So we have a dynamic mechanism, a quantum mechanism, to orchestrate across at least 12 or 13 orders of magnitude of scale, creating a multi-scale collective that is beyond merely chemical flows. I'm not disregarding all that great biology and all these action potentials, but I will say that this helps us to reevaluate the information processing that the brain can do at maximum. So if we look at 100 billion neurons operating at Hodgkin-Huxley, that's kilohertz, that's 10 to the 11th times 10 to the 3, we get 10 to the 14th ops per second. But if we now go to the picoscale, where these superradiance states are operating, the max computational rate is 10 to the 11th times 10 to the 12th, which then brings us to 10 to the 23 ops per second, which is beyond exascale, 10 to the 18th. Now, this also motivates, as many people have pointed out, a reevaluation of the energy consumption requirements of these systems. And they don't scale like classical digital computers. If we look at Frontier supercomputer, which operates at exascale, we have 20 megawatts. That's just a small fraction of a nuclear bomb, but 1,000 megawatts, right? And then the brain, operating at 6 orders of magnitude less power, can actually have more ops per second in a pulsed fashion. And that's, of course, less than a 60-watt light bulb. Now, how might this be? I mentioned those subradiance states, which can dynamically coordinate quantum state memories. And in fact, many quantum computing specialists are using those right now to actually manipulate quantum systems for long-lived quantum memories. And at the same time, we can use these superradiance state power outputs for error correction. And look at that. If I take 100 billion neurons and I multiply it by the power output of even just a single one of these filaments, this is a very conservative estimate, I get about 1 megawatt, which is on the order of the Frontier supercomputer. So what is the origin of the information processing power and capacity? I argue that it's superradiance. So just to point out here, 13.9 seconds, we're in numerical precision. So this is essentially, on the time scale of picoseconds, essentially infinitely trapped for its subradiant state. So I just wanted to point out that quantum effects are not always fast. Sometimes they're long, but they're long in the collective, not at the single molecule level. Okay, I wanted to give a short conscious interlude because I think it's relevant as we discuss how do we go from thinking of these architectures and structure and function to thinking about the computational capacity of life in relation to the universe, which is the title of this talk that was advertised. So I start from the presupposition that David Chalmers starts with in his book, Character of Consciousness, which is that not all truths are deducible from physical truths. And so I do say that strict materialism is therefore false. Why do I say that? A key premise that Chalmers doesn't make, but that I will as a physicist, is that even an idealized human intelligence, one that is aided by computational intelligence, AI intelligence, maybe another superintelligence, is still confined in the physical universe by the following: thermodynamic randomization, because we have insufficient memory, we have finite memories. We also have quantum uncertainty. Even if we could go beyond machine precision, we would then have, if infinite precision in one observable would predicate complete ignorance of a complementary observable. And then finally, the relativistic horizon of our observable universe. We are an open system, open quantum system, always unveiling new objects to us. So these all serve as quantifiable limits from 19th and 20th century physics on complete physical knowledge and unrestricted deducibility. And so we should think about that as we just have conversations about AI, that AI is as limited by the physical laws of the universe as human beings are. So I also want to point out that Newton's and Schrödinger's equations do not necessitate the structures and functions of any of these architectures that we're talking about: slime molds, brains, cats, water, galaxies. None of these things are built into those equations. Schrödinger equation is just unitary evolution caused by the Hamiltonian. There's nothing about initial conditions, but you have to add that on top of it. and we are sufficiently ignorant of the universe's initial conditions to actually predict all of these things a priori. Now, math also does not encode the quality of phenomenal experience. Consider C.D. Broad's mathematical archangel, another superintelligence that cannot derive ammonia's smell merely from its structure. Eigenvalues of the Schrödinger equation do not give us conscious phenomenal experience. So, and then from the spiritual perspective, in both the Bible and the Upanishads, the multiplicity of the universe is related to an underlying ordered reality. In Hebrew, dabar, in Greek, logos, in Sanskrit, Brahman. And the human subject with a soul, ruach, pneuma, or atman, survives even after the body decays. So liberation for the soul with an imperishable body is gained by recognition of, submission to, and oneness with the ultimate truth that is certainly not materialist alone. It's a state of conscious being rather than a mere statement of fact, which is what I'm giving to you in this talk. So Max Planck, one of the founders of quantum mechanics, I think would agree with me. And in fact, he has this great book called The New Science from 1959. Albert Einstein gave the introduction and called Max Planck one of the few scientists who entered into the holy of holies of science. You know? And Max Planck articulates that this dichotomy between human free will and deterministic causality is an illusion. In fact, it's a logical disjunction. In fact, that is resolved when you think of limited observers like us, limited observers like all things within the physical universe that have insufficient memory, finite access to available energy, and finite computational power, but then a superintelligence that exists outside of space and time, an ultimate truth, if you will, that then has clear-seeing spiritual powers of vision, in fact, a divine vision. So let me go from way up in the clouds now to back down to Earth. So here is a great article from the New York Times that came out at the beginning of March before my Science Advances was published. And it actually discussed this topic of mycorrhizal fungi and these networks of fungi that operate in trees, and they're doing computing dynamically underground and above ground. So my group has been really interested in a similar organism that is not mycorrhizal fungi, but is a slime mold, an aneural, single-celled, multinucleate organism. This is the fungi on the left that shows this kind of shuttle streaming behavior, which we also observe, and this is work with our collaborators at Tufts University, showing histological stains of Physarum polycephalum. This is a many-headed slime mold, but it's a single body with many nuclei and only one cell. And you can see here, they've got nuclei that are distributed throughout these networks. And at the front, you can see a number of actively metabolizing areas. And this is where you have very high ATP consumption relative to the other parts of the body. And this shuttle streaming behavior has been essential for understanding its computational prowess and its ability to solve problems like the traveling salesman problem. So here, this is work that my postdoc, Suyash Bajpai, has done in collaboration with our colleagues in Japan, Masashi Aono. But here you can see we take a stellate chip that we've custom fabricated, we take a projector, and we run a Hopfield neural network to instantiate a traveling salesman problem with light. So because Physarum is photoavoidant, it avoids certain wavelengths of light, we can dynamically control the light in each one of these lanes, and because there's 64 lanes, Physarum can actually solve the 8-city traveling salesman problem. It's been shown in prior works, in fact, that Physarum can solve the 4, 5, 6, 7, and 8-city TSP problem in near linear time, which is beyond our best classical heuristic algorithms, like the Lin-Kernighan heuristic. Now, this is not a proof because, of course, this is not an exact solution, but it is a highly optimal and high-quality solution that Physarum arrives at. And so what Suyash did was he analyzed the power spectral density of the solution lanes in Physarum and these non-solution lanes. And what we are seeing here is that before illumination starts, you can see that the two are rather indistinguishable. But then as you turn the optical feedback on, you have a downshifting of the energy to lower frequencies, and then a separation and amplification of the frequencies, amplitudes that are in the non-equilibrium steady state for the solution lanes. So this is what we think are signatures of the first organismal condensate, Froelich condensate, that we've seen in Physarum polycephalum. We're currently rebuilding this setup both at Howard in DC and in Japan, and so stay tuned for results as we extend this problem to larger and larger city sizes to see if Physarum is actually using a potential quantum algorithm to solve this problem, or quantum degrees of freedom, because Physarum has access to all of these fibers that I mentioned in the earlier part of my talk. So, in talking with cosmochemists and other people who are interested in looking at the interstellar media, this is a great article from 2018 in Physics Today that studied the polycyclic aromatic hydrocarbons. These are called astropause. And these are essentially large sheets of benzene that exist in the interstellar media and around circumstellar clouds. And interestingly enough, if you excite them with ultraviolet photons, you can lop off hydrogens, you can break carbon bonds, and then over time, you can actually produce tryptophan. And in fact, we've seen signatures of this on very ancient asteroids, even in our own solar system, that predate Earth. And so the question is, if these tryptophan networks actually hitched a ride on asteroids and became the seeds of these architectures that we see ubiquitously in protein architectures throughout biology. Okay, so now thinking about the stars, we then think naturally about the whole universe. So Margolis and Levitin in 1998 proved that physical systems have a minimum time required to evolve between two orthogonal states, that's 0 or 1. Now, even though we can use the Schrödinger equation and unitary evolution to do this, we can also show that in the macroscopic limit, and I do this in my science advances, that in this macroscopic limit, the result is only different by a factor of 2. So even for classical macroscopic systems that don't, let's say, obey Heisenberg uncertainty, you can still have this computational maximum that is defined by Margolis and Levitin. So just other assumptions that I'm making in this calculation going forward, I'm neglecting all effects that are outside of 3+1 spacetime, anything beyond our observable horizon, anything beneath the Planck scale, and anything involving dark matter and dark energy. So I'm strictly restricting to matter-based computation with the field. So I can neglect also geometric factors like 4/3 and pi for clarity of the exposition. So here I can then compute, and this was done by Seth Lloyd in 2002, who showed that the maximum ops of the matter-dominated universe is 10 to the 1/20th, using the rest mass energy of the universe. And then if I do this again, how much time do I have, Stuart? A couple minutes, good. So essentially what I can do is by taking, using Hubble's constant and by equating the galaxy near the horizon at r, moving away with velocity hr, I can actually relate this quantity to the whole age of the universe divided into chunks of the Planck time. And it's interesting that it scales as the square of this quantity, which hints at a holographic principle on the universe boundary. So I can do the same thing for the computational capacity of life, given the existence of these superradiant states that are actually controllable and operate above the thermal noise floor, unlike single molecule computations that may be within that floor. So this is the most recent census of carbon-based life on Earth from PNAS 2018. So I can take that value and the upper bound as set by these superradiant protein fibers, and I get the following estimate for all kingdoms of superradiant life on Earth. So if I equate the maximum computation that can have been performed by the observable universe since the very beginning to that value, I see a very interesting relationship. And this is the conjecture that I made in the science advances earlier this year, which relates both the Planck scale behaviors through superradiant life to the Eddington-Dirac large number, which is the ratio of the electromagnetic force to the gravitational force between an electron and a proton. So why is this interesting for quantum computing specialists? Well, in December of 2024, Google came out with the Willow chip. It's a lattice-based surface code that beats the break-even point. And that break-even point is something that these architectures in biology have been doing for a long time with error correction cycles that are on the picosecond scale, whereas Willow's chip is on the microsecond scale. So it's a really interesting phenomena that now we have these architectures of qubits that are millions of size, and the state of the art in quantum computing is now how to scale this break-even that Google has achieved, a remarkable achievement, and to scale it now for larger and larger systems. Maybe we can learn something from biology. Now, if you're afraid of a big bad quantum computer merging with AI to produce a supercomputing oracle, then I offer a warning, fear not, because in fact, if human beings have entangled qubits, we can actually verify truth claims and truth demonstrations that are beyond us to prove using our conventional classical computers. And so whether we do that using silicon or we use that using our own biology, this can be a way for human beings to cooperate and defeat any type of supercomputing oracle that emerges. This was from Quanta 2019. So in close, I just want to highlight some collaborations and the work that we're doing across these many scales, and in collaboration with colleagues at the University of Arizona and the University of California, Santa Barbara, and at NanoBioSim, the University of Waterloo, and of course Google Quantum AI, we are working to build a national center for exploring quantum aspects of life, where we'll look at all of these different questions in all their rich, many degrees of freedom. So finally, this is our standard model of biology. And as we look at these standard models of biology, we have to reevaluate them. And work that has come out of my group and others over the last 10 years has given us new insight to understand how we can push the bounds of physics as we re-examine some of the foundational assumptions of biology. And so I've talked to you about two works today, primarily in SuperRadiance, my Science Advances work, and also the work that we're doing in a neural slime mold. So here's a summary of what I've just told you, and here are those references, and I'm very happy to take any questions we have in the time remaining. Thank you so much for your time.
Thank you, Philip. That was awesome. That was awesome. We have time for one or two questions while our next speaker, Anita, sets up. Please raise your hand. Kind of hard to see up here.
I think there's one there.
Yeah, she's got it.
Thank you. That was a wonderful talk. I've just got a few basic questions. I think I know maybe some of the answers, but one of the things that tryptophan and other aromatics do is in different media, they will change their spectral absorption. Have you tested for that? I expect you have, and the curve that you showed suggests that there is an effect with the length of the microtubule. But how would you respond to that? And also, what about other aromatic amino acids? Should we be looking for other kinds of networks in them?
No, very good question. So the first thing is, yes, in aqueous solution, tryptophan is better modeled as a 3-level system. The 1LB state relaxes very fast to the 1LA state on the order of tens of femtoseconds, and that changes in different environments. But on the time scales that we're measuring, which is picosecond as well as nanosecond fluorescence lifetimes, those are relevant. So it's a 2-level system essentially. Now, your other question about, you had asked about other aromatics, right? So they're much less fluorescent than tryptophan, so the effect will be much less. But tyrosine, for example, congregates around tryptophan and gives, by Forster resonance energy transfer, a lot of its absorption strength to tryptophan. So tyrosine kinds of funnels over to tryptophan. Phenylalanine also absorbs, and these aromatics like cysteine also absorb. And there are other sort of modalities that you could have superradiance in those wavelength regimes that are not in the absorption maximum of tryptophan. So yes. Yes.
Any more? here real quick and then...
If microtubules emit superradiance as tryptophan absorbs UV photons from mitochondria, for instance, if the tryptophan in the microtubule are performing the sort of information processing that leads to consciousness, could any of the superradiance be related to heat emission required by Landauer's principle during the performance of computations?
So I went very quickly to this, but if you look in my science advances, that's exactly the argument that I take. And in fact, if you take these superradiance states, integrate them over the whole brain, in fact, you can show that obeying Landauer's principle, you have error correction rates that are perfectly tuned thermodynamically. They don't overheat the brain, and they actually do very well energy throughputs that aren't going to tax the brain beyond its rest mass energy. So in fact, exactly right. We can use Landauer's limit to compute how much heat is erased as the brain error corrects. And so using all these quantum degrees of freedom, you're going to need lots of error correction. And in fact, that's what the brain has. And it's got an inbuilt classical coprocessor that can read out this information. Yes.
Great. Let's thank Philip for a wonderful talk.
Thank you. Thanks, Stuart.
Our next speaker is Anita Goel, the Chairman CEO of Nanobiosim Research Institute, speaking on quantum information processing and nanomachines that read-write DNA. Does physics need a revolution to explain life and consciousness? Anita Goel.
Thank you. Oh, thank you. Good morning, everybody. It's a pleasure to be here today. I want to thank Stuart for inviting me to join you today, and also for his long-term dedication to making the field of consciousness a pursuit that so many of us are passionate about turning into a hardcore scientific pursuit. So thank you, Stuart, for your dedication. So I am going to share with you a bit of a journey on looking at our biological systems, such as DNA, nanomachines that read and write DNA. Are they processing information quantum mechanically? Do they have non-trivial quantum mechanical effects going on? And what can we learn from these kinds of systems about the bigger question that we've been asking about is our physics complete? Is quantum mechanics complete, as Roger Penrose has articulated? So we'll play with that, and hopefully through an interactive process, we will take that a little bit forward. So as Stuart mentioned, I'm the chair and CEO of Nanobiosim, which one part is a research institute that works at the nexus of physics, biomedicine, nanotechnology, information science. We also have a spin-off company that's commercializing digital diagnostics. And one of the sort of lifelong quests that I've been very interested in is when you bring fundamental physics together, including classical and quantum mechanics, an understanding of life in living systems, and information processing on very small scales. At that nexus, what can we use with that multidisciplinary approach to sort of attack the problem of consciousness? What you see here is rural Mississippi, where I grew up. Spent a lot of time meditating in this nature-type environments, wondering about the big questions of the universe. And sort of the deeper I went in my study in physics and medicine, places like Stanford, Harvard, and MIT, I became more aware of how disjointed in our modern framework understanding of physics and living systems is. Most of our physics was developed in the last century in the context of inanimate matter, which were closed systems operating at or near equilibrium. It hadn't really come to terms with life, living systems, and consciousness. And most of our medicine today is practiced at the level of chemistry and molecular biology. We haven't really integrated how physics plays a role in fundamental physiological processes in our practice of medicine, whether it be mechanical forces, electromagnetic fields, quantum effects. And so I was inspired to see that Einstein, towards the end of his life, came to a similar conclusion, that one can best appreciate from a study of living things how primitive physics still is. And about 100 years ago, Albert Einstein gave us a framework to try to see how matter and energy are interconnected, popularly known as E equals mc squared. And I came to the conclusion that in order for our physics to come to terms with life, living systems, and consciousness, we're going to need an equally, if not more profound, expansion of our theoretical physics framework to not just put matter and energy on some kind of equivalent footing, but bring matter, energy, and information, and even this thing called consciousness, in some kind of interplay, interfooting. And we have to understand at a physical level, not only how matter and energy interact, but how matter, energy, and information talk to each other. And even this thing called consciousness, which we as a scientific community need to define how that plays with matter and energy. So I needed an experimentally grounded system to sort of explore these edges of physics and not get only lost in theory. So about 20-some years ago, I fell in love with this thing. These are little nanomachines whose job in life is to Xerox DNA. They read and write information stored in a DNA molecule. Now, there were techniques becoming available from quantum optics to actually experimentally probe these molecules at the single molecule level. Now, most of our traditional biology has been developed in the context of ensembles of molecules, hundreds and billions of molecules averaged over their behavior to understand, for example, chemical kinetics. So here's a way that I began diving into and how to study their real-time dynamics at the single molecule level. Now, I hypothesized that this DNA is like a piano, and these little enzymes that read DNA are operating and reading and writing information in such a way that they're talking to many layers of information that are embedded in the environment. In other words, the music that you as an organism make is an interplay of not only the information stored in your DNA sequence, but information that's stored in the environment. And this little nanomachine is processing information in a very remarkable way, not only processing what's in the DNA base pair, but the whole environmental milieu. Now the question is this a classical dynamics, or can there be quantum mechanical effects that are informing how this nanomachine reads and writes information in DNA? Are these dynamics purely classical, or is there quantum effects being exploited? And that's what I will walk you through. Both a theoretical and experimental commingled journey to try to attack this problem. And then the other issue is, if you think of things like evolution, is evolution purely random, or could information in the environment inform evolution? So I hypothesized that at least at the molecular level, evolution might be more Lamarckian than Darwinian, where information in the environment might be talking to and influencing how this little motor enzyme reads the DNA. So keep that in the back of your mind as we go. So I had spent a sabbatical at the Santa Fe Institute studying different kinds of biological and other kinds of networks, and came up with a way to think of these nanomachines as algorithmic state machines that are going through these internal states, internal conformational states, and cycling through them to determine their external translocation along the DNA, and more about that in a second. So in my research institute at NanoBioSim, we have been working with a number of government agencies who are not interested so much right now in the fundamental physics of life and consciousness questions, but are very fascinated about how we can harness these nanomachines and exploit them, whether classically or quantum mechanically, for various kinds of control, and how we can use them as new kinds of sensors. And so from genomic sensing to biochemical sensing, all the way to quantum sensing and computing, if these nanomachines are truly processing information in their environment in a quantum mechanical way, well, why can't we harness them in some sort of hybrid devices that can do quantum sensing and quantum computing? And in our lab and spin-off company, we have built capabilities to take some of these deep insights and design principles and harness them in engineered devices like microfluidics, MEMS, and nanofluidic systems to turn some of those capabilities into devices and products. Here's an example of something that our company has built and gotten FDA approved, which is something called, where it does nanosensing. It harnesses the ability to read DNA and amplify DNA with very high precision using these DNA polymerases, and do what typically takes a big fat 400-pound machine and a handheld mobile device in a fraction of the time. So what if we could quantum control these nanomachines that read and write DNA at the single molecule level? What would that mean? Well, for one thing, I think Roger Penrose and Stuart would be very happy, because it would give us new tools to explore some of these fundamental questions at the foundations of quantum mechanics and even the edges of quantum mechanics in the context of open systems that are living systems, and this search for new physical principles. Some more applied things is, could you do quantum information storage and computing with this kind of architecture? And some, for those enthusiasts in the room, how quantum mechanics might play a role in molecular evolution. Could there be some sort of entanglement between the environment, locally and non-locally, and how DNA information flows through the medium of DNA, which is a self-replicating system. So here's some work that kind of summarizes the experimental understanding with our theoretical kind of classical model, at least for this explanation, how as you tune the tension on the DNA string with one molecule and you measure the rate at which the little motor reads the DNA or replicates the DNA, we find that we can tune that speed. As you stretch the DNA, you slow its replication rate. And then around 35 piconewtons, you come to a screeching halt. And if you keep pulling, you can get the motor to go in reverse gear and start unzipping DNA, which is its normal error correction machinery that we have triggered by the external mechanical force. So one of the editors at Scientific American got excited about this, so we got this little cover art talking about how you tune DNA strings. And this is an example of what we can do in our lab, as well as what the theory is going after. So we can take a molecule of DNA, stretch it out with lasers, and visualize in real time at the single molecule level how a little nanomotor replicates, converts single-stranded DNA into double-stranded DNA. And we can do it as a function of the tension that we're putting on the DNA, the mechanical stress. And we find that as we increase the tension, we actually slow this process of replication. And if we increase it even more, then we can actually stall the motor. It goes into this kind of limbo state where it doesn't go forward or backwards. We've halted replication. And then if we pull it even harder, we can start to unzip the DNA and start reversing the process of replication. And this is just the tip of the iceberg of how other kinds of information that are embedded in the environment, that are supposedly non-biological, could be talking to and influencing this information processing of this nanomachine as it's reading and writing the DNA. So here's a book that I did a chapter with, that Roger Penrose did the foreword, and I was inspired by my friend Paul Davies. We were having a number of conversations about this. So here we start to examine the first of all, can these nanomachines plausibly be having quantum mechanical effects? Now, before I show you more stuff, I just want to start with the very fundamentals. Now, the typical reason why people believe that biological systems will not have quantum effects because they're wet, warm, and swampy, we think everything will decohere, there won't be enough coherence that makes it relevant to have quantum mechanical effects. So in the paper, I summarize this heuristic calculation, which in a nutshell, don't have time to get into this right here, but happy to talk about it afterwards. The fundamental time that it takes for a motor DNA complex to decohere has to be larger than the critical timescale it takes for a little motor to read one base pair of DNA in order for quantum effects to be relevant on that scale. And so the typical time it takes for a motor to read one base pair is about 10 milliseconds, and the estimated decoherence time can be anywhere from minutes to hours, depending on the modes of relaxation of a long polymer. So that just goes to show heuristically that quantum mechanical effects are plausible. Now the question is, how could quantum mechanical effects enhance DNA nanomachine function and efficiency? Well, one is in the area of energy transduction. It turns out these nanomachines are highly efficient in converting chemical energy in the form of DNTPs into mechanical work. Whereas your car may be 20 to 40% efficient, these machines can be 99.9% efficient. Also, in the area of quantum information processing, it has been speculated that they may use a quantum search algorithm to find the matching nucleotide from a large sea of candidates. There's a nice paper that actually Stuart sent me about base pairs as qubits, where the DNA base pairs are in a sort of qubit state between their normal and their tautomer, and they're in a quantum superposition of those states, and therefore you get an inbuilt quantum error causing a mutation rate. And also the switching from the forward gear to the backwards gear. This could be exploiting quantum effects when it goes from copying to error correction. And what I will talk also about is there are layers of information in the environment that could be coupling into the real-time dynamics of this nanomachine. And if we have the experimental resolution, can we start to measure a signature of that information in the environment into this real-time dynamic signal? So, as many of you might know, the 5 sort of criteria for a quantum computer are the DiVincenzo criteria, and so I argue there that this system meets all the requirements of being a quantum computer, including that the decoherence times can be much larger than the gate operation time. You can define well-characterized qubits. A colleague of Roger Penrose, John Barrow, had taken Eugene Wigner's quantum clock inequalities and applied it to a black hole and used it to derive the accuracy and precision of a black hole as the maximum lifetime, which is the maximum time a black hole can run, if you will, and be accurate, like a quantum clock in measuring time, is the Hawking lifetime of black hole, and the length scale of that would be limited by the Schwarzschild radius. And likewise, these Wigner inequalities put a quantum limit on the precision of a quantum clock, and applied to a black hole, this became the fastest possible information processing frequency. So in that paper, I applied this same Eugene Wigner quantum clock constraints to looking at a DNA nanomachine. It's meant to be for any measuring device. And here, if you plug in the parameters, this is again heuristic for a DNA polymerase. The theoretical estimate is the accuracy of this clock would be about 380 seconds, and the DNA polymerase makes an error about 1 every 100 seconds, at least for that species. And the precision, which is the sort of minimum time resolution that clock can register, if you plug in the numbers, turns out to be about a femtosecond, which is comparable to the lifetime of an internal transition state between one of these internal nodes on this network. And if you just use that, we estimated the computational, if you take the ratio of the maximum running time to the minimum running time, you can get the maximum number of computational steps. And if you see this while the motor is reading about 10,000 bases, we estimate again that this little nanomachine is making about 100 billion computational steps for every base that it reads. That's a lot more information processing that you might need to read just one base at a time. So you have well-characterized qubits that can be measured and operated on, and we can think of this, these internal cycles. Just to be clear, on this network, I don't think this pointer works, but the red pentagon on the right side is the polymerization cycle. This is when the motor, think of a Turing machine, is going from n to n plus 1. It's making one cycle in the red pentagon. And when it's going from n to n minus 1, it's making one circle in the green pentagon. And when it's doing nothing, it's in the black triangle. And so we modeled that this nano machine, when it's moving forward, it's going from the nth state to the n plus 1 state, it could be in a quantum superposition of these internal states, much like the DNA tautomer. And this is where it could be exploiting quantum coherence in some of its dynamics. And so before I show you some experimental data, I want to show you, I'm going to come back to another theoretical framework we have been using to sort of extract more information about how this system behaves. So let's go back to Feynman. He had this very nice analysis of the Ratchet paradox, Ratchet and Paul paradox. And the idea for a long time was that could you exploit thermal noise and fluctuations to create directed mechanical work for free, like a free lunch? That was the basis behind this idea of perpetual motion machines. And the idea goes something like this. You have this Ratchet and Paul in a thermal bath, and you have Brownian motion particles hitting that little windmill-looking thing. And every so often, it'll start rolling, and because you have a ratchet and a pawl, you'll get some directed movement of this gear, and the pawl will hold it in place. So this would be like a rectifier, where you've rectified random Brownian noise into directed mechanical work. So people are like, great, we can turn that into a perpetual motion machine. We can somehow extract energy from the thermal bath and turn it into directed mechanical work. And Feynman explained very nicely how you can't violate the second law of thermodynamics, because you have to maintain detailed balance, but you can get something. You can't get a free lunch, but you can get a cheaper lunch if you drive the system out of equilibrium. If you put these two temperature baths at different temperatures, you can harness thermal noise in a rectified way to help your energy transduction efficiency. It's not free. You're still feeding it energy, but you're being more efficient in the conversion of chemical energy or one kind of energy to another, transducing that into work. This goes back to the idea of a Maxwell's demon. Now I want to come back to something that we've covered in this conference. This can be Brownian noise that gets harnessed into mechanical work, or harnessed into some kind of directed energy motion, but it can also be quantum noise, so zero-point energy. The same mechanism could apply as a mechanism to tap into harnessing quantum zero-point energy and rectifying that for some kind of use. So think about that as I show you the next slides. This goes back to the classic Maxwell's demons problem, which is, if I have a system in equilibrium, can I drive it out of equilibrium? The idea, you have this little demon that can sort molecules. The low-speed molecules go one way, the high-speed molecules go another way, and you intelligently separate it from equilibrium to out of equilibrium. And again, that would involve something that would violate the traditional second law of thermodynamics. So now, in a very experimentally grounded way, I wanted to map our system into can we exploit this ratchet-like picture to get new insights on how thermal or quantum fluctuations could be harnessed and exploited into enhancing the function and efficiency of these little nanomachines. So I hope you're with me here. So this is the DNA crystal structure of the DNA polymerase. You see it has a polymerase and exonuclease domain. And this is the traditional chemical kinetics mapped over 10 years of traditional biological biochemistry experiments over bulk ensembles that measure these equilibrium constants and transition rates. And this is the traditional kind of chemical kinetics picture. And this is now me turning it into like a network picture where each node on the network, there's a topology here that's defined by those allowed interstate transitions, and each node represents an internal conformational state that there's some experimental data for, either from structural or chemical kinetic data. And we use as a starting point these internal rate constants that have already been mapped in ensemble to model kind of on average what the single molecule behavior would be. But then again, there's a lot more randomness and stochasticity when you look at the single molecule level that gets averaged out when you look at it at the ensemble level. And this gives us a very nice model to understand how different knobs in the environment can couple to and be used to sort of modulate or tune how this dynamics flows through this network, both externally, macroscopically, but also internally through these internal transition states. And what I then do is then map that same energy, that same lattice, so the same network, into a 2D lattice where we separate mechanical movement along the DNA railroad track, n to n+1 on the x-axis, and put the chemical reaction coordinate on the y-axis. So as you can see, as the polymerase goes from n to n+1, it's moving along the black axis, and this red is the hydrolysis of nucleotide energy conversion that's happening as the enzyme moves one cycle in the red pentagon. And it's work it has to do to incorporate one base pair. And from X-ray crystallography, you have some of these internal states that have been visualized. So we're trying to ground it very deeply in many types of experimental data. Now you think of this 2D energy landscape, this lattice, which also looks like a 2D energy landscape, as a way in which, and now you have, as you're doing the ATP or NTP hydrolysis, this energy landscape is shifting, and you're getting a ratchet-like phenomena that is driving this forward. Now, there's two ways to think about this. One is thermal noise, where you have thermally activated barrier crossings that enable sort of thermal noise to be rectified in a way that improves the energy conversion efficiency or the function of this nanomachine system. But another way is quantum. So you could have quantum tunneling effects, which tunnel through these barriers. You could have a number of kinds of quantum effects that are effectively also creating higher efficiency in this ratchet-like mechanism of rectifying Brownian and quantum fluctuations, thermal and quantum fluctuations, into the directed movement in this network. So I have designed in our lab an experiment where it's the equivalent of like a double slit experiment where we would try to distinguish definitively, is there a quantum mechanical effect? Or if you're in a superposition of these internal states, we would expect to measure an interference pattern that's shown on the right side. If it's truly classical, and we made a measurement of this position over time, multiple times, we'd expect it to converge along one of those yellow dots. So that's similar to the double slit experiment, where if there's an interference phenomena, you see this as you do a bunch of experiments, you see this probability distribution, whereas if it's classical, everything clusters in the way on the yellow dots. And so this is a very clear way of getting a definitive answer in these very complex kinds of experiments on whether quantum mechanical phenomena are going on or not in this system in a way that is both theoretically and experimentally testable. So in our lab, experimentally, we have just started to get those signatures. We don't have the resolution yet to make that measurement. But just to show you how these experiments themselves are very complex, but we can measure the position versus time of the nanomachine as it's reading along the DNA at different forces, or piconewtons. And this is kind of the noisy signal that's a little bit more averaged. And then as you start to take the derivative of that, what we find is that the motor doesn't move like your car on cruise control along the highway. It actually moves in little stochastic bursting gaps. And I believe that in that fine structure dynamics of these so-called stochastic bursting gaps is a signature of that information that's embedded in the environment. And as we improve the resolution with which we can tease that out, this gives us a window into mapping that matter-energy information interplay, but in a hardcore experimental way that we can measure and quantify. So this is the motor, for example, running in Xerox mode and in editing mode, forward gear, reverse gear. That's the position versus time. And if you take the derivative, you can see the stochastic bursting gap pattern. And you see there's different kind of noise going on here. Now, we have to improve the signal to noise even in these experiments, which is not trivial, to be able to even get at some of these other features that are from the environment. But we have some interesting ideas on how to play with the environment to tease out some of these effects. So this is how I think I've covered this. I'm going to come back now full circle. So we've heard a lot from, for example, Roger Penrose, that quantum mechanics is incomplete. In fact, Einstein and Schrödinger complained of the same thing. And the question is, how do we get to a more complete theory of quantum mechanics? And more broadly, how do we get to a more unified theory in physics that helps to reconcile quantum mechanics, general relativity, and makes physics compatible with life, living systems, and consciousness? I believe that we really need to understand how matter, energy, and information are connected, and from information also understand how that connects to consciousness. And in order to attack the consciousness problem, by the way, I think we need to take a both theoretical physics and experimental approach. So in a very grounded way, we can map out this new physics. And I want to leave you with this story. When I had the pleasure of spending a sabbatical at the Santa Fe Institute, I used to have lunch with Murray Gell-Mann many times, and we would engage in these different kind of debates and on consciousness and other big foundational questions of physics. And I asked him one day, I said, do you agree with the following definition of consciousness based on your work on complexity? And he agreed. And that consciousness is some kind of emergent phenomena that emerges when matter attains a critical degree of complexity. It begins to manifest self-awareness and consciousness. So that is almost some of the prevailing scientific paradigm, which is very much based on a matter-based paradigm. So I provoked Murray Gell-Mann and asked him, well, what if we could rewrite all the laws of physics to be consistent with an alternate paradigm where information and consciousness are something fundamental to the fabric of reality, and matter, energy, and space-time emerge out of that? And what if those new laws of physics are compatible with the current laws of physics in one limit, but give us new insights into phenomena that we haven't been able to measure or detect yet? Because if you don't have a framework to try to even understand those things, you won't design the experiments to try to go look for those things. So how our worldview is shaped affects our theoretical frameworks, our models that we make, which then affect in turn the experiments we design and the information we glean back out of them. So I'll leave you with that. I'd love to take questions, comments, and conversations. Thank you.
Thank you. All right, a few questions over there. Who's got the mic? Hang on. Third row over there.
Thank you for the speech. Do you have an opinion how DNA can read the environment?
Do I have an opinion on how DNA can read the environment? Yes, I do have an opinion.
What's
okay?
your opinion, of course?
So I think that, you know, you have the traditional view, even in modern science and medicine, is all your information is stored in your DNA sequence. You can have two people who have the same oncogene that causes a certain kind of cancer. One may get cancer and one may not. They have the same DNA sequence, but the environmental information is different that may turn on or off that gene. So it's a complex interplay about the information in the DNA and the information environment. I think that the information in the environment is on many layers. You may have the more trivial layers like biological transcription factors, but like we're showing mechanical forces, I believe it can be extended to things like electromagnetic fields and other kinds of effects, quantum effects, that could be coupling into this and informing it. So you ask a big question, we can ask like evolution. Did this all happen randomly, or is there some information that is coupling and sort of guiding the arrow of evolution? And I would argue that at least at the molecular level, evolution might be more Lamarckian than it is purely Darwinian.
Thank you.
Yes.
First of all, I want to thank you for the wonderful presentation and wonderful work that you're doing.
Thank you.
It was amazing. I want to go to the last slide that you shared.
Yeah.
And I 100% agree with you.
Yeah.
And I think that's what's missing in both physics and science today.
Yeah.
That from particle to go to such wonderful things,
Yes.
it cannot be coincidence.
Yes.
So in the evolution here, it must come from consciousness to all this. And all the evolution have got also will or intention. And this is something which is missing in physics, the will and intention. And only when it's come from your assumption of the last one, that consciousness derives this, only then we can stand there is will and intention and love in the universe. Thank you.
Yes, and it's a major paradigm shift, because if we don't even think in that framework, we won't even construct a theoretical physics model to even look for the experiments to find that. So we can be blissfully unaware just because we didn't think that way. Yes.
Great talk, Anita. Thank you for that. I was struck by the relationship of mechanical stress, the tension, in relation to the volume of information that was derived. And I'm thinking of this in terms of tubulin as kind of a tensegrity structure. So was there by slowing the nanowires down, you were deriving more information?
So I'm not making any comment on the amount of information. I'm just saying that the speed of the little nanomotor that's reading the DNA that we measure at the single molecule level, that is slowing down when you increase the tension. And as you increase it beyond a certain point, it reverses gear and starts to run backwards. So it's a knob in the environment that's tuning the entire dynamics of how the little motor is reading the DNA.
Any other questions?
Yes, back there.
Beautiful talk, thank you. I was wondering how symmetry is involved in your strategies, because symmetry is somehow related to beauty and performance. Did you see symmetry in your work, and where?
Yeah, so that's a great question. So I think that there is underlying, again, this is now an opinion, there's underlying mathematical structure and information and symmetry in that layers of information that is embedded in the environment. And that information starts to trickle down into these living systems that are very open. They're talking to their environment constantly. They're exchanging matter, energy, and information. So what I'm trying to do is, through these very complex experiments, try to see if we can tease out some signature of that underlying information symmetry or structure, that we could get an experimental trace or signature. That's not noise. And this is, I don't want to make this sound easy, this is hard, right? But this could be like a holy grail. Can we actually see that information playing a role in matter and energy in a way that we can quantify, understand, and map out this new physics triangle between matter, energy, information? And what's interesting about living systems is they're also technically conscious systems in our traditional understanding. So if we start to understand this thing called consciousness, we can also understand how it interplays with this stuff. So this is, again, just the beginning stages of trying to map that out.
Okay, let's thank Anita for a fantastic talk. Okay, our third speaker is Travis Craddock from the University of Waterloo, and he will tell us about magnetic effects in quantum neurobiology. Travis.
Thanks, Stuart. All right, can everyone hear me okay at the back? All right, good. All right, thank you, Stuart. Starting off, I'd like to thank the conference organizers for inviting me here, allowing me to speak at this plenary session, allowing me to set up a workshop on Sunday, if you happen to attend that, as well as to give a concurrent talk. I'd also like to acknowledge my team at the University of Waterloo. Have only been instantiated there for about a year, so we're growing. We're making strides in this area of quantum neurobiology. And I'd like to point out specifically the work I'm going to be talking about today has been primarily headed by my research associate, Dr. Zeta Hagigi, and my master's student, Caleb Sigüenza. I'd also like to thank my collaborators that I've been working with since joining the University of Waterloo, particularly Dr. Robert Smith at Nova Southeastern University and Christoph Simon at University of Calgary, as well as the funders who have funded and supported this work. So just to give you an overview of what I'm going to be talking about today, I'm going to start off with an introduction of how I view quantum biology, what I define as quantum biology, and then talk about the quantum brain hypothesis. I'm sure we've heard about it a lot this conference, but I'm going to go into it again and give my unique take on it, and then talk specifically about how I look at the quantum brain hypothesis in relation to the kind of work I do at the University of Waterloo. So the relation of quantum biology to quantum neurobiology and neuroinflammation for the purposes of finding novel treatments and diagnostics for neuroinflammatory illnesses. I'll then go into talking about the neuronal cytoskeleton. Again, we've probably heard a lot about microtubules at this conference, but I'm going to give you an in-depth view of the neuronal cytoskeleton and what its function is. And then talk about weak magnetic fields in biology in general, and then focus directly on weak magnetic field effects on the microtubule cytoskeleton and some experimental and theoretical results that we have currently ongoing. Okay. So quantum biology, we've heard about it, we've talked about it all morning. We've heard about quantum effects, quantum physics, and looking at the difference between quantum physics and classical physics. Specifically, what I'm talking about in terms of quantum biology is looking for non-trivial quantum level effects at the level of molecules, atoms, and subatomic particles that have an influence upon biological function. Now, there's two main branches of this area of quantum biology. There's using quantum effects to study biology, and that's commonly referred to as quantum for biology. So there's a lot of technologies that are coming out to use quantum principles, light, magnetic fields, other nanotechnological devices to study biology and learn more about it. I'm in the other camp, looking specifically for the ways that biology may take advantage of quantum effects to give life an edge, to give life something unique in order to improve its efficiency or improve its evolutionary capabilities. In general, this area of quantum in biology, which I'm talking about, there's generally three main areas that have been looked at. One we've heard about from Dr. Kurian this morning is light-matter interactions, and primarily this has been looked at in terms of photosynthesis, so plants' ability to harvest energy from the sun and convert it into usable chemical energy, as well as vision, so the way that the human retina can pick up single photon events. Another area is looking at electron and proton transport within biology, so things like enzyme catalysis, ion channels. The way that those biological structures function is they have to take advantage of the movement of these subatomic particles, so an electron or a proton. And because we're dealing with subatomic particles, the best way to describe it is in terms of a quantum level event. And so trying to understand how those processes take place at the quantum level is very, very important for the way that biology actually works. Specifically, one that I think is primary to look at is the electron transport chain in mitochondria. This is where all of the energy of your cell comes from. It relies very much heavily upon the electron transport chain, and trying to model that from a quantum level description I think is a very important process. And then finally, kind of the focus of the talk, is going to be magnetic field effects in biology. So there's been a growing work looking at specifically weak magnetic fields and how it influences the way that biological systems function, both at the cellular level as well as at the gross macroscopic scale. And the prime example of quantum biological magnetic effects is what's known as the avian compass or bird navigation. So looking at how birds, migratory birds specifically, how they can travel long distances without visual cues, without any other cues, but know exactly where to go. And the idea is that these birds have this built-in ability to sense Earth's magnetic field, and they use that for navigatory purposes. Now, there's ideas of little tiny magnets in the brain, but the leading candidate from a quantum biological standpoint is built on radical pair mechanism inside the retina of a bird's eye, and I'll get into that in a little bit. Okay, quantum brain hypothesis. Now, we've heard about it here at the conference, but when did this first come about? And I'm going to state that it came about almost initially as soon as the ideas of quantum mechanics came out. So this is Erwin Schrödinger, a father of modern quantum mechanics, and he wrote in his initial essay, What is Life?, where he talked about how quantum effects could potentially influence living systems. He has a section where he specifically talks about what this means for brain function and what it means for consciousness. And he followed this up in an additional essay called Mind and Matter. So the idea of quantum effects playing a role in our cognition, the way that our brain works, and in consciousness goes all the way back to the initial inception of quantum mechanics. But I'll argue that the popularization of this idea did not come about until the theories of Penrose and Hammerhoff in the late 1980s and early 1990s. And this idea really ingrained in the public consciousness, this idea that quantum effects could play a specific role in conscious observation and the way that our brain works. And we've heard a lot about supporting evidence here at the conference. But when this initially came out, the idea of the brain appealing to quantum level events below neurons, within the submolecular realm, that when it first came out, it was met with fierce criticisms by many people. Max Tegmark argued that the brain was too warm, wet, and noisy to support quantum phenomenon, and we've heard about that from Dr. Goel's talk, that this is a common it's a common argument against quantum biology, but it's a little bit of a misconception. Because more and more as we study these systems, we learn that the warm, wet, noisy environment can actually afford a degree of advantage to these quantum systems that's not found in completely isolated, sterile quantum physics systems. So I'm not going to go into the details of that, but it's one argument for or against quantum biology. The other was raised by Christoph Koch, who's been at this conference before, and he argued that there's no need to appeal to the level of quantum level events to understand brain activity. We just need to look at the level of neurons and up. That's complicated enough, and consciousness arises from the complexity of these neurons interacting, so why bother appealing to these low-level events? And then there were some philosophical reasons brought forth by Patricia Churchland. There's been many rebuttals, many arguments, and this has spurred a lot of scientific research in this area. And so despite the criticisms that were initially raised against this idea, it has really driven the idea of looking for quantum mechanisms in the brain and defined a field of quantum effects in the brain. We've got great work, we've heard from Dr. Kurian, Dr. Goel, looking for quantum level effects and how it affects brain processes, even much so that the harshest critics of this idea have now proposed their own ideas of quantum mechanisms within the brain. Okay, when we come down to the kind of work that I'm looking for, I want to take this idea of quantum effects in the brain and apply it to neuroinflammation. So what is neuroinflammation? Neuroinflammation is inflammation like any other inflammatory response. You bang your arm, you get a bruise, it becomes swollen, it becomes warm and hot, and that's a normal process that your body uses to protect the injured area. It then goes through a process of cleansing and cleaning out the area and eventually repairing it back to its regular state. The brain has its own specific neuroinflammatory response, and it's no different except that the fact that the brain is isolated from the remainder of the peripheral body. So in this inflammatory response, if you get an injury in your head, whether it's physical due to some sort of mild traumatic brain injury or a concussion, whether it's chemical due to some neurotoxic exposure, or whether it's biological due to some sort of viral infection, there's certain things that go on that accompany this neuroinflammatory response. And one is the increase in oxidative stress, and this is characterized by an increase in reactive oxygen species. They're oxygen molecules that are highly reactive, that are constantly looking for extra electrons, and they steal them from all over the place, and this can cause a lot of damage within a system. As well, the carefully balanced metal ions within the biological system, within the brain, become thrown out of balance. And because these metal ions are very, very tightly regulated, this can cause a host of problems. Just like in Alzheimer's disease, there's dysregulation in iron and zinc. In Parkinson's, there's a dysregulation of iron and copper, and so forth. So a lot of these mental and neurological diseases have this imbalance of metal ions stemming from inflammation. In the short term, this imbalance of reactive oxygen species, ROS, and metal ions is neuroprotective. But if it's long-lasting and chronic, this is where it becomes a problem. This is where it becomes a slow degrade of the biological function of the neurons and something that causes long-lasting damage in neurodegenerative diseases and neurological disorder. Now, in neurodegeneration, we heard from Stuart the other day that in things like Alzheimer's disease and Parkinson's and Huntington's disease, it stems in part from the destruction of the microtubule cytoskeleton, which holds up the cell. But somehow this relation between how the cytoskeleton is destroyed and how neuroinflammation plays a role, there is some sort of link, there's a correlation, but we don't know the exact mechanisms of what's going on. Now, I'm arguing that in order to understand this, we want to look at specifically how things like reactive oxygen species and the way that they steal electrons, those subatomic particles, is best understood from a quantum level description, or understanding how those metal ions interact with the dysregulation of those metal ions interact with the cytoskeleton is best understood from a quantum level perspective. And that's the goal of my lab. Okay, so the neuronal cytoskeleton. The cytoskeleton is a protein structure made up of the tubulin protein, which is shown over here. So this is tubulin. It's made up of two heterodimers. One is alpha tubulin in light gray here. The other is beta tubulin. They're always found together within the cell. And then these tubulin proteins self-organize and stick together to form long strands called protofilaments. And these protofilaments then stick side to side in a slightly staggered pattern and wrap around to form this hollow tube called a microtubule. Now, this hollow tube is very structurally stable, and it forms long processes, and it can hang around in a cell to support the shape and structure of the cell. It's found in all eukaryotic cells, from your skin to your heart, to your liver, to the brain. Now, in the brain, it's very specific because it has to hold up the very unique architecture of the neuron. So it forms these bundles that Philip was talking about earlier to support the axons and dendrites of the neuron, and they're very stable over time. Whereas in regular cells like kidney cells or heart cells, they're very labile and they move around a lot, and they form radially out from the center of the cell. They're also the things that are responsible for pulling apart chromosomes during cell division, in dividing cells like other cells within your body, whereas in non-dividing cells like neurons, you don't get that effect. Now, this process of self-organization of microtubules, of tubulin forming together to form these microtubules, is a GTP-dependent process. And so as tubulin sticks together and forms this long cylinder of a microtubule, it hydrolyzes this GTP to release some energy, and it allows the microtubule to grow. If there is a lack of GTP and all that GTP gets converted into GDP, and there's none of the GTP left around, what will end up happening is that the microtubule will spontaneously fall apart if there's no other stabilizing proteins. And there's this kind of process that goes back and forth of catastrophe and rescue, of growing and shrinking that naturally accompanies microtubule formation. But in neurons, this is stabilized by stabilizing proteins like tau and MAP2 that keep them from falling apart, even in the absence of GTP. In things like the synapse of the neuron, these microtubules form distinct architectures. They deliver motor proteins throughout the cell, so little motor proteins, kinesin and dynein, walk along the surface of the microtubule to bring cargo from the cell body all the way to the distal regions of axons and dendrites, where they then place things like vesicles full of neurotransmitters for release in signaling, where they place things like the ion channels for picking up and receiving signals. They also stabilize the mitochondria, the powerhouses of the cell, and shuttle them around the cell. So they're very, very important to the overall function of the neuron, not just specifically for the structure, but also for the function, because they do play a role in synaptic signaling. They control, to a degree, the strength of the synaptic connection, as well as the firing rate or the ion transfer rate of ion channels. This is the same in dendrites. So these microtubules actually go up into the dendritic spines, and they change the length of the dendrites. So you might have heard of synaptic plasticity. That is the changing of the shape of these dendritic spine shapes. And the microtubules very much control that as they polymerize and depolymerize. So let's come back to quantum biology. So how does quantum biology play a role in microtubule function? We heard earlier from Dr. Kurian about light-matter interactions. This is some work that we've been doing. I've done some in my lab. I've done some in conjunction with Philip's group. And then we heard about Philip's work on SuperRadiance. There are unique optical properties of microtubules, both in the SuperRadiance regime as well as the energy harvesting and energy transfer regime. And these are actively being studied in multiple labs around the world. Philip's and myself being two main ones that are represented here. But that's not what I'm talking about today. I do think it's very interesting and something that we need to look at further. But what I want to talk to you today about is specifically about magnetic field effects. Okay, so there is evidence for weak magnetic field effects in biology. The evidence for weak magnetic field effects covers a broad range of areas. So one, it's been shown that weak magnetic fields, and when I talk about weak magnetic fields, I'm talking about things on the order of maybe 5 millitesla or below. So if we're talking about things like MRI or fridge magnets or anything like that, we're talking on the scale of Tesla and up. So I'm talking about 1,000th of that or below. And so what they've shown is that weak magnetic fields can influence cognition and memory. So there's been initial studies looking at applying weak magnetic fields to the brain, and they've shown that it can improve memory tasks and cognitive-related abilities. However, there's no known reason why this would occur. In addition to weak magnetic fields, there's also effects in biology related to isotopes in biology. So in this regard, an isotope is a version of an atom where the electrons all remain the same, but you're changing the nucleus by adding in an additional neutron or removing a neutron. And it slightly changes the weight of the atom, but one of the things that it can also change is the nuclear spin of that atom. And so the nuclear spin, you can think of it as a little tiny bar magnet inside the center of the atom, which can then respond to magnetic fields, or it could then be influence some of these other weak magnetic field processes. So things that have been shown are things like lithium-6 and lithium-7, which differ by a single neutron, have been shown to have different effects in animal behaviors. So this is work that's been suggested or pointed to by Matthew Fisher in his quantum brain hypothesis based on Posner molecules. There's also been supporting work by Zoya Leonenko's group at the University of Waterloo, where she has shown that there are differential effects in neuron firing patterns in the presence or absence of lithium-6 versus lithium-7, as well as in, I forget what the other ones were. but definitely within neural firing patterns in the presence and absence of lithium-6 and 7. It's been also shown that anesthetic potency of xenon, which is a single atom anesthetic, the potency of xenon will change depending on which isotope you look at. So just by changing the number of neutrons within xenon, you can change the amount of anesthesia required to put a patient under. And they showed this in rats, and they varied the xenon isotope. And xenon has several stable isotopes with varying nuclear magnetic moment. And they found that as you increase the magnetic moment, that you would require more xenon anesthesia to put the animal to sleep. And finally, there's been work out of Russia that has shown that by changing magnesium isotopes, magnesium is required for ATP synthesis. By changing the magnesium isotope in ATP synthase and applying or removing a weak magnetic field, you can increase or decrease the rate of ATP synthesis. So these are just a handful of experiments that point to this idea that weak magnetic fields play a role in biology, but we have no mechanism. There's no real strong evidence for a mechanism of why this may occur. And one of the reasons for that is that from a classical standpoint, the nuclear spin of an isotope or these weak magnetic fields, which are very, very small, they cannot be explained by classical physics because the thermal energy in the environment is much stronger than the energy that would be elicited by these weak magnetic fields. So why is it that you're seeing an effect from these weak magnetic fields, even though just random motion around you should completely wash it out? And so what they suggest, how this could occur, is through a quantum mechanism known as the radical pair mechanism. So the radical pair mechanism is the leading candidate for the explanation of weak magnetic fields and magnetic isotope effects within biological systems, in general in any chemical system, but specifically in biological systems. So in the radical pair mechanism, you have two partners in a chemical reaction coming together. Blue and orange come together in a cell or in some sort of environment. And in this case, one of them acts as a donor and gives an electron to the acceptor. And in doing so, you now have two unpaired electrons. And those unpaired electrons, they also have a spin associated with them. And depending on the way that electron is shared, those spins can be in opposite directions, shown over here on the left, which is referred to as a singlet pair. So this is now a singlet radical pair. Or they can be pointed in the same direction. And this is what's known as a triplet radical pair. And depending on how those are pointed, whether they're both up or both down, or some sort of combination of up and down, there's 3 different ways to form them to give rise to this triplet. That's why it's called a triplet, whereas there's only one way to get this singlet. That's why it's called a singlet. You have this singlet triplet radical pair formation. And in reality, what actually happens is that they don't exist as either singlet or triplet. There's some sort of superposition between the two, and these kind of oscillate back and forth. Now, over time, the singlet formation will allow it to rapidly convert to the end chemical product, and you get the end of the chemical reaction, and you go on your merry day. Whereas the triplet reaction, it's very, very difficult for it to recombine, and therefore it's a very, very slow transition to end up at that end product. Or, because of the way that the triplet electrons are formed, it can sometimes go off and form some sort of alternate products, and you don't even complete the chemical reaction you were looking for. Now, because there's a difference in rate between the way that the singlet and the triplet radical pairs end up at the chemical end product, you can then try and influence this chemical reaction through external influences, either through the presence of a weak magnetic field or through the presence of some sort of isotope that has a nuclear magnetic spin. And by influencing it, you can try and force it to stay in the triplet state, which would mean that chemical end product it takes a much longer time for it to come about, or you can force it to be in the singlet state, which means you can speed up the process. And this is a way to influence the chemical reaction rate and drive the biological system. And this is exactly what is proposed to occur in the birds in order to pick up a magnetic field. So the leading theory of the avian compass is that within the eye of the bird, in the retina, inside of your rod cell, inside the rod cell, between the two discs, there's a protein called cryptochrome. And this cryptochrome molecule is light sensitive. And it's really close to several tryptophan amino acids within the protein. And what happens is that a particle of light comes in, it excites this flavin molecule within cryptochrome, and that excitation causes it to enter into a chemical reaction where the excited electron on flavin is shared between flavin and the tryptophan molecules. And then it enters into this radical pair. And when there's a weak magnetic field, it can prolong the time that it's in this excited state, or it can, in the absence of it, it can drive it so that that excited state drops off fairly rapidly. And depending on which state it's in, it will change the way that cryptochrome is behaving, and the way that cryptochrome behaves then influences whether or not the rod cell fires and sends a signal through the optic nerve back up to the brain. And it's through this that they believe that the birds can sense the magnetic field. This is something that's actively being studied. There's experimental support for this. It's an idea that takes a quantum biological effect based on radical pair chemistry and ties it directly to neuron firing and overall behavior. And so our thought was, if this can occur in something like cryptochrome, could it occur at another location within a brain-relevant protein? And specifically, we're talking about the cytoskeleton, because that's what I like to research. So coming back to this idea of cytoskeletal polymerization, like I said, tubulin, when it polymerizes, it takes advantage of a molecule called GTP. There's two GTP molecules within tubulin. One is hydrolyzable, it can convert to GTP, and one cannot. And so this conversion takes place in the presence of a magnesium atom. Now, as the microtubule grows, there's a cap of GTP on the end of the microtubule, and you can think of it like the plastic on the end of your shoelace. As long as it's there, the microtubule won't fall apart, just like your shoelace won't fall apart if the plastic is there. But as the GTP is hydrolyzed, if you remove all of that GTP, then the microtubule will fall apart. If you remove the plastic on the end of your shoelace, it frays. It's the same sort of idea. And so the idea is that if there's some sort of radical pair that could be formed or influenced by this magnesium ion here, we may be able to influence the rate at which GTP hydrolyzes or does not hydrolyze, and then control the amount of tubulin that's polymerized in a system. And so that's what we wanted to do. We wanted to polymerize microtubules in the presence of magnesium isotopes in the absence and presence of a weak magnetic field. And we used 3 different versions of magnesium, which are all stable. Magnesium 24, which is primarily the most abundant magnesium that you find naturally within any system. It has 0 nuclear spin. And then there's a heavier version of it called magnesium 26, which also has 0 spin. And then there's one right in the middle of middleweight called magnesium 25 that has a 5 nuclear spin. So we can rule out any sort of mass effect because we're going to look at both light magnesium and a heavy magnesium, and then also look at a magnesium that's in the middle that has this nuclear spin effect. So what we did was we set up Helmholtz coils. These are electric coils which we pass current through. It sets up a stable magnetic field in between the 2 coils. And then we took plates of tubulin in tubulin polymerization buffer, put them inside the magnetic coil, and then polymerize them in the presence of this weak magnetic field. And we changed out the buffers such that we could change whether we had natural magnesium, magnesium 25, or magnesium 26. And then we measured the amount of tubulin polymerized using an optical density assay. So we shine light through it, and because microtubules scatter light, we could see how much light was passing through and get a measure for how much of the tubulin is actually polymerized. And so these are our results. So these 2 here are natural magnesium. This here in the middle, this is magnesium 25, and this is magnesium 26. The blue is when there's no external magnetic field applied. There's still Earth's weak magnetic field there, but nothing applied. And then when we apply a 3 millitesla magnetic field with the Helmholtz coils, that's what's shown here in pink. And what we see here is generally there's no real statistical difference across any three isotopes in the presence or absence of a magnetic field, except for magnesium-25 in the presence of the magnetic field, and we get a gross increase in the amount of tubulin that's polymerized by about 4 to 6 times what we would normally get in the absence of the weak magnetic field and magnesium-25. So we're seeing statistical difference. We're ruling out anything due to a mass effect because, again, we use that light magnesium and that heavy magnesium. And we want to make sure that we are looking specifically at this mechanism, this radical pair mechanism. So we want to make sure we're ruling out anything else that can occur in a system. Because microtubular polymerization has been shown to be dependent upon pH of the system. It's been dependent on temperature of the system, and it can be quite finicky depending on what the external conditions of the environment are. So first and foremost, we want to make sure that we're not doing any sort of heating to the system. When you're applying current through these Helmholtz coils, they can warm up. And therefore, we can expect potentially that we're going to get some sort of temperature gradient across the plates. So one thing we did was when we first set this up, we made sure that we only put tubulin within wells that were far away from the Helmholtz coils to try and minimize any sort of heating effect. We then measured what the temperature was in each well and tried to correlate that with changes in the polymerization density at the end of the experiment, and we found absolutely no correlation between temperature differences and optical density changes. And therefore, we can rule out that this is not due to a temperature variation across the plate. We then wanted to make sure that it wasn't due to slight variations in the magnetic field. Now, when we studied this, the accuracy at which we could measure the magnetic field was down to about 0.02 millitesla, and we measured where the magnetic field was, and it's fairly consistent across the plate. But to ensure that we weren't seeing something due to slight differences between the wells, between runs we would cycle where the different conditions were on the plate. So if one was up in the upper left-hand corner, magnesium-25 was at the upper left-hand corner at one of the runs, the magnesium-26 would be up there at one of the other runs, and the magnesium-24 on one of the other runs. And we did this multiple times to try and average out any effects that may be due to changes in the magnetic field effect. And we found no correlation, again, between changes in the magnetic field and measurements of optical density. And so we conclude this is not due to a field variation effect. So then we came to the final point. Okay, so now we see this effect. We do believe it's due to the magnesium isotope and it's due to the magnetic field. Is it consistent with a radical pair? So people have measured this type of thing before in other systems, but they have not tied it directly to a radical pair mechanism. And so Hadi Zedekigi, very talented research associate in my group, devised a radical pair mechanism. We were agnostic as to what the radical pair actually is, and we had a general setup of what the radicals could be with several parameters, and we ran a machine learning optimization to try and identify which parameter sets would match our experimental results. And we found a small subset of theoretical results that would match our experimental results. And from that set of parameters, we can kind of start to deduce what the radical pair may be involved. Okay? And that's something we're still currently active under investigation trying to figure out. But from these predictions, things that we found is that in the presence of magnesium-25, you get this gross effect due to the presence of the nuclear spin of magnesium. But even in the absence of the magnesium-25, you're still getting an effect of the weak magnetic field on the polymerization rate. And so you can see that here when we zoom in close to the level of Earth's magnetic field. So Earth's magnetic field is 0.05 milliteslas, which is located here. And even as we scan, slightly above that and slightly below that, we're getting increases and decreases even in the absence of the magnetic magnesium isotope. It doesn't look like much, but this value is 1 and it's a ratio, so this is 100%. Whereas if you drop down slightly, you're dropping down to about 50%, so half of the tubulin being polymerized. If you go up a little bit higher, you're getting 20 or 30% higher, just by varying slightly around the Earth's magnetic field. That is obviously exacerbated in the presence of this magnesium-25 isotope. So, I'm saying that the radical pair mechanism is the leading candidate to explain weak magnetic field effects in biology. I've presented some evidence for that, primarily drawing on the work of the avian compass to point to that description and the experimental results that they've come about. This has been established, like I said, in cryptochrome, but it remains to be seen if it's viable in other biological conditions. And I'm proposing it here specifically for microtubule polymerization. Our results indicate that microtubule polymerization is sensitive to weak magnetic fields, and that is exacerbated in the presence of magnesium isotope effects. And this is purely a quantum radical pair mechanism. So you cannot explain this in terms of any sort of classical based behavior. Now, what does this mean? Okay, so we have these nice experimental results. What does this have bearing on? And two things that come to mind are, one, there's a potential for this to provide some sort of weak coupling between neurons within the brain. So within neurons within the brain, you have axons and dendrites which are in close proximity, and every time you have a neuron fire, there's ions flowing along with the action potential, and every time there's a movement of ions, movements of charged particles generate magnetic fields. They're generally very weak. However, this mechanism is sensitive to weak magnetic fields, and therefore, at least in a close proximity, you would have firing neurons can then couple to other neurons by changing, microtubule reaction rates or polymerization rates. And it's a potential mechanism that can provide some sort of coupling, similar to ephaptic fields, but this is not electric fields extending over the brain. This would be magnetic fields which are in closer proximity to the firing neuron. This also has implications for cognition and memory. So like I said, there's experimental evidence that weak magnetic fields can influence cognition and memory. And as we heard from Stuart's talk, by modulating microtubule strength, we can improve cognitive states related to Alzheimer's disease. And this may be a mechanism in which these weak magnetic fields are improving the cognition and memory in these experiments. Additionally, and this is kind of an interesting area, is that all of the experiments that we do, except for some that are in isolated systems, isolated from Earth's magnetic field by special metals called mu metals, but in general, everything on Earth is within Earth's magnetic field. And so biology, I believe, has evolved to optimize itself to be functioning around and using Earth's magnetic field strength. But when we leave Earth's orbit and we get outside of Earth's magnetic field, then there's a lot of biological processes that could be affected by the absence of Earth's magnetic field, this being one of them. And so it's been known that for extended periods of space travel, that there have been certain health issues that astronauts have faced. This may be one of them. And so I would propose, looking at long-term cognitive effects of space travel to ensure that we're not increasing chances for neurodegenerative diseases or any other sort of thing that would rely upon stability of the microtubule cytoskeleton. So that being said, I've listed references throughout my talk, so you can find them here. I know it's small. If you can't get a picture of that and you can't read it, then contact me afterwards, and then I'll open it up for questions. Thank you very much.
Okay. Questions? Back there first. Oh, is that not the mic?
Hello?
Oh, we got over here.
Okay, sorry.
Hi, Travis. I have a very weird but potentially supportive data point to what you're saying. And also, I may be mispronouncing his name, but the time crystals guy, Nestor Mercado, there's something he said that stuck with me. And in his talk, he said, halos are real. So this is just weird, but 2 years ago, I got really sick. I had all these symptoms that resembled multiple sclerosis. I had over $20,000, $30,000 worth of medical bills, MRIs. And during the worst week of it all, I had a lot of weird animal encounters, and 2 of them with birds. And in one of the cases, a bird actually flew and hit me in the head. So these doctors, they didn't know what it was. They think it was long COVID in the end. But someone had put me on a lot of magnesium. And I don't know what I'm talking about, but I was taking like excessive amounts of magnesium as well. So I was feeling my head like buzzing all week. So like, I'm not even kidding you. Like a bird literally flew and hit me in the head. That's never happened before in my life. So it rhymes with all this magnetoreception stuff that you're talking about, and that's it.
So I'm not sure. I'm not sure how that would relate, but thank you for the anecdote.
Next question. Second row there, please.
Thanks for a great talk, Travis. I'm curious about whether there's any indication of how these magnesium-25 ions or isotopes might occur in certain places, either in parts of the brain or within neurons.
Yeah, so natural magnesium is primarily made up of magnesium-24. It's about 80% magnesium-24, which has the zero spin. Another like 11 or 12% is magnesium-26, which is the other zero spin magnesium isotope. And then there's about 8 to 10% that is magnesium-25, and that naturally occurs, and that's throughout your body. And so the distribution throughout your body, I would say at any given time, is roughly 10%. As naively approaching this. There has been other studies, not with magnesium, but looking at other isotope fractionation within biological systems. And in certain cases, they find that sometimes the biological system prefers to hang on to some of these rare isotopes of a given chemical. I don't know if it's calcium. I don't quite remember what it was, but there are fractionation studies in biology to show that biology will hang on to some of these rare isotopes. I don't think anything's been done to look at that in terms of magnesium in biology in general, or specifically the brain, but I think it would be an interesting area to look at.
Thanks so much, Travis, for the nice talk. We've talked a little bit about how magnesium is useful in hydrolysis for lots of proteins, enzymes, DNA repair enzymes, as Anita was talking about in polymerase. Have you thought about how the object, I know you were agnostic about it in your slides, but have you thought about how magnesium co-locates in these sort of cages with, and this could be the radical pair mechanism, and then the other is what would be the photoexcitation stimulus, like analogous to cryptochrome, and what is the wavelength regime there?
Okay, so yes, so we have thought about magnesium in terms of other enzymatic processes. So the work of Buchachenko in Russia, he looks at ATP synthesis, which is an analogous mechanism to this. We take the agnostic approach because he proposes that magnesium itself is formed in the radical, but there's issues with magnesium oxidative chemistry where that's not very likely. So some of the other players, I've been talking to people like Peter Hoare and Jonathan Woodward, some of the other players like hydroxyl groups and so forth, may or may not be long-lived enough to support a radical pair mechanism, and that's why we took the agnostic approach and why we're trying to narrow it down. So right now we're trying to be agnostic and follow the evidence where it may lead. In terms of excitation, this mechanism does not require an excitation. So for example, in the avian compass, the light comes in and that's what sets up the electron to jump up in orbital, and then it's shared between flavin and tryptophan. And that's what sets up the radical pair. In this case, it's the hydrolysis itself, because you're changing the chemical bond. There's a period there where electrons are being moved around, where the radical may be formed transiently. And so we're not quite sure if it may not need a photo excitation. However, because we're agnostic, there could be a photoexcitation process. In that case, it would have to be either from a biophoton, or in the case of our experiment, it may just be due to the ambient light within the lab. We did not do the experiment in total darkness, and it's something, again, we're proposing to look at.
It would raise questions about these classical microfields that are generated in neurons by ion flows, which could then stimulate local magnesium effect spin radical pairs along the neurons or proteins or whatever.
Yeah, this is very interesting. Yeah, we'll talk about it later. Yeah, for sure.
Any other questions? One more right there.
I noticed that in your experimental setup, the orientation of the magnetic field was axial in relation to the sample of cytoskeleton protein. Have you seen if the effect is attenuated if you change the orientation to being peripheral, or any of the three possible orientations?
Are you reviewer two on our paper?
Pardon me?
Are you reviewer 2 on our paper?
No,
Because that's exactly what they asked. They said, did you account for orientation? No, we did not. The way that the microtubules are forming you got the little wells, and yeah, the magnetic field is cutting across the well this way.
Yes.
But the way that the microtubules form, it's amorphous at that point when it's just within a dish. If we were to apply a electric field such that the microtubules would align in a direction, we could start to measure the effect of the orientation of the magnetic field comparable to the orientation of the microtubules. But that, again, is work that's still ongoing.
That's precisely my point, because if you're not seeing a change in the take-up, that points to a quantum effect rather than it generating from any sort of a Maxwell effect or field effect.
Right, yeah, exactly. Yeah, so we want to look at if there is sort of orientation effects that.
Thank you.
Okay, let's thank. Let's thank Travis, all 3 speakers. Great session. See you at 11:10.